final common pathway of catabolism of glucose, amino acid, and fatty acids. Glucose, amino acids, and fatty acids. It occurs within mitochondrial matrix. TCA cycle is aerobic. Why TCA cycle is aerobic? And this TCA cycle is intimately involved with respiratory chain. The respiratory chain is cannot operate without oxygen. Oxygen is a must for operation of the for operation of the respiratory chain. I mean, also for the operation of the TCA cycle. Okay, when oxygen is absent, TCA cycle stops. TCA cycle cannot be operated. The TCA cycle provide energy. It also provides building blocks for synthesis of some important amino acids like glutamate, glutamine, aspartate. The first product of the cycle is the first product of the cycle is citrate. Is it clear? The first product of the cycle is citrate, and citrate has three. Is citrate? It has three carboxylic groups. It has three carboxylic groups. It has three carboxylic groups. That means citrate or citric acid has three carboxylic groups. So it can be termed as tricarboxylic acid cycle. We are not going into details of the chemistry, but if we see, if we analyze this citrate or citric acid, we find citrate has 6 carbon, 3 carboxylic, it has 3, it is a citrate, it has 3 carbon, 6 carbon. Of these 6 carbon, 2 comes from acetyl-CoA, 4 comes from oxaloacetate. Oxaloacetate and acetyl-CoA condense together to form citrate, and citrate has 6 carbon, and it has 3 carboxylic groups. 3 carboxylic groups. So it can be rightly termed as tricarboxylic acid. Okay, tricarboxylic acid cycle, TCA cycle. We also ask in the exam why TCA cycle is called so. Why it is called tricarboxylic acid cycle? It is called tricarboxylic acid cycle because the first product citrate has first product of the TCA cycle, citrate or citric acid has 3 carboxylic groups. That's why it is called tricarboxylic acid cycle. Okay, TCA cycle. It is also called as simply it can be termed as citric acid cycle. Okay, or Krebs citric acid cycle according to its discoverer. Mr. Krebs discovered two important cycles. One is citric acid cycle, another is urea cycle. So if you say Krebs cycle, the meaning, it means dubious meaning. You must mention Krebs citric acid cycle. Okay, so citric acid cycle has three names. One is citric acid cycle, another is a Krebs citric acid cycle according to the name of its discoverer, Mr. Krebs, and TCA cycle, tricarboxylic acid cycle. Tricarboxylic acid cycle is called because it has carboxylic, three carboxylic group. TCA cycle is called amphibolic pathway because of the simultaneous operation of both anabolic and catabolic reactions in the pathway. The catabolic reactions is acetyl-CoA is completely oxidized in TCA cycle, yielding carbon dioxide, water, and energy. Is it clear? And that energy is used for the energy is produced, which is in respiratory chain, ultimately used for synthesis of ATP. Okay, so it is catabolic because acetyl-CoA is completely oxidized in the TCA cycle. It is anabolic because intermediate products of the TCA cycle, as for example, succinyl CoA, alpha ketoglutarate, succinyl CoA, oxaloacetate. These are the uh, intermediates in the TCA cycle. This, uh, these are used for synthesis of some important biomolecules like heme and heme. Succinyl CoA is is the building block for synthesis of heme, and from heme, hemoglobin, myoglobin, cytochrome oxidase can be synthesized. Okay, alpha ketoglutarate is the product which we will be soon viewing the this is citric acid cycle. Uh, alpha ketoglutarate is used for this is used for synthesis. It is also a intermediate of TCA cycle, and it is also used for synthesis of glutamate, glutamine. Glutamine. These are the non-essential amino acids. These are amino acids, non-essential amino acids. Succinyl CoA is used for synthesis of heme. Okay, oxaloacetate is used for synthesis of aspartic acid, which is again non-essential amino acid. Is it clear? This upper the view glucose converted to pyruvate. This is termed as glycolysis. Oxidation of glucose to pyruvate can be termed as glycolysis. The product of glycolysis is pyruvate. Pyruvate or lactate. Pyruvate in aerobic condition, it is this pyruvate enters into the mitochondria where it is where it is converted to acetyl CoA, and acetyl CoA is completely oxidized in the TCA cycle. When oxygen is not available, pyruvate do not enter into the TCA cycle. It do not enter into the mitochondria. It is simply converted to lactate. This is this here this portion. When oxygen is absent, it will be converted to lactate. It can be seen from this view, or the earlier view. This pyruvate converted to lactate in anaerobic condition. So we are again concentrating on TCA cycle. Very important steps of TCA, important steps of TCA cycle or outline of TCA cycle is important. It is very easy to remember. Entry of pyruvate into the mitochondrial matrix. Uh, the enzymes of TCA cycle are within mitochondrial matrix. So the reactions of the TCA cycle takes place within mitochondrial matrix. Oxaloacetate and acetyl CoA condense together to form citrate. Citrate. This oxaloacetate is a 4-carbon compound acetyl-CoA, two carbon compound, this together, condense together to form citrate, which is six carbon compound. It is also called tricarboxylic acid because it has three carboxyl group. Citrate ultimately oxidized to isocitrate and isocitrate and then alpha ketoglutarate. Is it clear? Alpha ketoglutarate. Here, one thing you should remember, when pyruvate converted to acetyl-CoA, pyruvate is three carbon compound, acetyl-CoA is two carbon compound. Naturally, there is loss of one carbon. This one carbon is used for synthesis of carbon dioxide. Carbon dioxide. So one molecule of carbon dioxide is produced in this step, during the conversion of pyruvate to acetyl-CoA. Again, another molecule of carbohydrate could be produced when isocitrate is produced. Isocitrate is converted to alpha ketoglutarate. These are reactions as carbon dioxide is produced, carbon is coming out. This is called decarboxylation reactions. Okay, oxidation and decarboxylation takes place simultaneously. And this, when isocitrate converted to alpha ketoglutarate, also when pyruvate converted to acetyl-CoA. Alpha ketoglutarate is converted to alpha ketoglutarate, then converted to succinyl-CoA. Succinyl-CoA is a four carbon compound. Another molecule of carbon dioxide is produced here. Okay, is it clear? Succinyl-CoA is converted to succinate. Here, one molecule of ATP is produced. That means GDP is converted to GTP. That means ATP is produced. Okay, GTP is an equivalent of ATP. Is it clear? Succinate is converted then to fumarate. Succinate converted to fumarate. Is it clear? And this all succinyl-CoA, succinate, fumarate, malate, oxalate, all four carbon compounds. All are four carbon compounds. Then again, succinate is produced from succinyl-CoA. It is succinate is converted to, next step is fumarate. Fumarate, is it clear? Fumarate is converted to malate. Malate ultimately converted to oxaloacetate. Is it clear? Then here one thing is required to remember. Ultimately, the citrate in citrate, the oxaloacetate oxaloacetate is has been used up. This citrate again regenerate when this oxaloacetate is produced. This oxaloacetate is produced, and it can be also produced by from carboxylation of pyruvate. This pyruvate is a three carbon compound. Oxaloacetate is a four carbon compound. When one molecule of carbon is added to pyruvate, there will be formation of oxaloacetate. That will be considered in gluconeogenesis. Up to this, so source of oxaloacetate is uh, either it can be regenerated in the TCA cycle, or it can be synthesized newly from pyruvate. Okay. So this is a short outline of TCA cycle. Now we can go a bit in details of TCA cycle. You see the first reaction, pyruvate, conversion of pyruvate to acetyl-CoA. this reaction, conversion of pyruvate to acetyl CoA has been detailed here. Conversion of pyruvate to acetyl CoA. Pyruvate is a 3-carbon compound. Acetyl CoA is a 2-carbon compound. 1 carbon is used for formation of carbon dioxide. Is it clear? The reaction, the enzyme responsible is pyruvate dehydrogenase complex. Coenzyme involved is NAD. NAD is oxidized form of NAD is converted to reduced NAD. Is it clear? This conversion of NAD to reduction. This is reduction conversion of NAD to NADH. It is the reduction reaction. This reduction is accompanied by oxidation of pyruvate to acetyl CoA. Okay, also with loss of 1 molecule of carbon used for formation of carbon dioxide. Pyruvate can now come to the point. Pyruvate dehydrogenase complex. It is called a complex because actually it is an aggregate of 3 enzymes and 5 coenzymes. Okay. Number of participants is also less. Okay. Again, come to the point. Enzymes. Do you here follow me? Do you follow?
Yes,
Yes,
sir.
sir.
Students? Yes. Okay.
Yes, sir.
It is a bit. You should follow patiently. Pyruvate dehydrogenase complex. It is called complex because it is an aggregate. Aggregate, you know, that means addition. It is a complex of 3 enzymes. Pyruvate dehydrogenase. Is it clear? Its coenzyme is thiamine pyrophosphate, which comes from vitamin B1, dihydrolipoyl transacetylase. Its coenzyme is lipoic acid, and coenzyme A. Coenzyme A comes from vitamin B5, okay, pantothenic acid, and dihydrolipoyl dehydrogenase. Its coenzymes are FAD, flavin-adenine dinucleotide, and nicotinamide adenine dinucleotide. So these 5 coenzymes participate in this reaction. 3 enzymes and 5 coenzymes participate in the reaction. That's why it is called pyruvate dehydrogenase complex. Okay. Suppose if uh, deficiency of these enzymes is very rare, if there is deficiency of one coenzyme, say this thiamine pyrophosphate, that means vitamin B1, if it is deficient, the reaction will be slowed. The conversion of pyruvate to acetyl CoA will be slow. That means there will be less energy production. It actually happens so in very, which is characterized by deficiency of thiamine. Your percentage is being given. Okay, so don't worry about percentage. Let me discuss this. Thiamine is characterized by deficiency of thiamine pyrophosphate. Now understand this biochemical defect can lead to energy deficiency, weakness. Others can be this FAD, NAD. This also comes from vitamin B complex. So vitamin B complex is very important for metabolism of carbohydrate. Is it clear? That's why doctors frequently prescribe vitamins, vitamin B complex. Next step we follow the This is the step oxaloacetate. This step simulate with this oxaloacetate acetyl CoA together form citrate, condensed together to form citrate. Okay, this FAD in this reaction, in this reaction, this citrate synthase is the enzyme. Citrate is converted to citrate is oxidized to alpha ketoglutarate. Also, there is decarboxylation. One molecule of carbon dioxide is produced here. The coenzyme involved is NAD. Is it clear? NAD is converted to NADH. Then alpha ketoglutarate is converted to, this is oxidized to succinyl CoA. One molecule again, there is decarboxylation. Another molecule of carbon dioxide is produced, and this succinyl CoA is 4-carbon compound, and the coenzyme involved is NAD. NAD is converted to NAD, reduced NAD. Okay, is it clear? The succinyl CoA is converted to succinate. Enzyme responsible is, again question comes, the alpha ketodehydrogenase complex. This alpha ketoglutarate dehydrogenase, alpha ketodehydrogenase complex is also a complex of 3 enzymes and 5 coenzymes which have been mentioned in PDH. Is it clear? It is this alpha ketoglutarate and pyruvate dehydrogenase complex. These are called complex because they have 3 enzymes, 5 coenzymes. Okay, and succinyl CoA is converted to succinate. Here, one molecule of ATP, that ATP equivalent, that is GTP, is produced. GTP ATP is almost same, just name a bit different. The GDP, this ATP is produced, or GTP is produced at substrate level. This is the energy that is produced directly in the TCA cycle. All other energies you see that could be produced when the reduced coenzymes are oxidized in the respiratory chain. Okay, succinate is converted to fumarate. Enzyme responsible is succinate dehydrogenase. The FAD is reduced to form this reduced FAD, then fumarate, malate. Malate, fumarate, these are interconvertible. That means fumarate can be converted to malate, malate could be converted to fumarate. This is very, so only some isomerase is required. This is not so important. This is bidirectional pathway. Malate, also a 4-carbon compound. Fumarate, 4-carbon compound. Malate is also 4-carbon compound. This malate is oxidized to oxaloacetate. Enzyme responsible is malate dehydrogenase. Coenzyme is NAD. NAD is NAD is reduced to NADH. Okay, oxaloacetate, then which is regenerated, it again reacts with another molecule of TCA cycle to produce citrate, and citrate again rotates this again, the repetition of the cycle is again the citrate. When it is produced, it is again there is this, in this way this is repeated. This oxaloacetate, it is acting in fact like a enzyme, not being wasted, and it condenses with new citrates to form, to operate this, for operating this TCA cycle, producing energy. This oxalates we mentioned can also be formed from carboxylation of pyruvate, that is not shown here. because it will be complex. So this is not for your view. Now come to the point, hexose monophosphate shunt will proceed as far as some as more as possible. Hexose monophosphate shunt pathway. The names, other name. We ask what are the names of hexose monophosphate shunt pathway? Name the other the what other names hexose monophosphate shunt pathway has? Pentose phosphate pathway, 6-phosphogluconate pathway, direct oxidative pathway. These three names it has in addition to hexose monophosphate shunt pathway. HMP shunt pathway, where it occurs? It takes place in cytosol. No ATP is directly consumed or produced in this metabolic pathway. Is it clear? Is it clear? Okay. This when glycolysis occurs, 90% of glucose 6-phosphate is oxidized in the Embden-Meyerhoff's glycolytic pathway. 10% of glucose 6-phosphate oxidized in the hexose monophosphate shunt pathway for production of ribose sugar, for production of ribose. And this ribose, not not only ribose and NADPH. Ribose is used for synthesis of nucleic acid, nucleotide, nucleoside, and nucleic acids. Then that means DNA, RNA. And the NADPH is used for of reductive synthesis of fatty acid, cholesterol, fatty acid, cholesterol, steroids, bile acids, nitric acid is also. It is also responsible for maintaining the reduced state of glutathione, which is essential for antioxidant activity, phagocytosis by WBC. This reduced energy is required, and it is also required for drug metabolism. Now come to the hexose monophosphate shunt pathway we mentioned occurs in the cytosol. Its sites of hexose monophosphate shunt pathway. Liver is the most important organ, adipose tissue, adrenal cortex, gonad, and erythrocytes. These are the sites where hexose monophosphate shunt pathway could take place. This is some important outline of hexose monophosphate shunt pathway. You see, the glucose 6-phosphate is the substrate for hexose monophosphate shunt pathway. 6 molecules of hexose monophosphate, 6 molecules of hexose 6-phosphate, 6 molecules of glucose 6-phosphate decarboxylated to form 6 molecules of ribulose 5-phosphate. That means this ribulose 5-phosphate, this is the keto sugar, and it has this 5-carbon compound. So 6 molecules of carbon dioxide is produced. Okay, and 6 molecules of ribulose 5-phosphate produced. This is pentose, and from this basic pentose sugar, there is synthesis of 2 ribose 5-phosphate, 4 xylulose phosphate. This ribose